Hello and welcome to this webinar titled End User Perspective on Setting in Process in Endotoxin Limits, hosted by Biopharma Asia magazine and presented by Dr. Friedrich from Wintzingerode from Roche Genentech. My name is Orsolya Balog and I will be the moderator. Before we begin, I would just like to remind our viewers that there will be a live Q&A session at the end of the presentation. Audience members can send their questions at any time during the presentation via the Questions tab located at the right-hand side of your screen, and we will go through these at the end of the webinar. Now, please allow me to introduce Dr. Friedrich von Wintzingerode, our speaker. Friedrich studied biology with focus on microbiology at the Technical University of Braunschweig, Germany. He received his diploma at German Strain Culture Collection in Braunschweig, Germany, and a degree at Institute of Microbiology and Hygiene in Charité, Berlin, Germany. Since 2001, he has been employed at Roche Diagnostics, working as micro-lab manager on various topics. In his role as senior manager microbiology, Friedrich led several Roche Genentech micro-expert teams, for example, identification and microbiological testing. He is the head of Roche Genentech Global Endotoxin Expert Group since 2013 and Roche Genentech Global SME on microbial product contaminations. Friedrich will be doing the presentation titled End User Perspective on Setting in Process in Endotoxin Limits. Welcome, Mr. Wintzingerode.
Thank you, Ursula, for the kind introduction. And I would like to start my presentation by giving you a brief background about biologics and how biologics are produced. So large-scale production of biologics, and if I talk about biologics, I may have to explain or define this. I define this as recombinant therapeutic proteins, like, for example, monoclonal antibodies. And the production, the large-scale production, consists of two steps, which in many cases occur at different production sites. The first step is drug substance manufacturing. That means the manufacture of the active pharmaceutical ingredient, in this case, or as an example, the monoclonal antibody. The second step, the drug product manufacturing, that means the manufacture of the final drug, the formulated monoclonal antibody filled in syringes or vials, for example. And again, those two different production steps in many cases occur at different production sites, sometimes in different continents. Let me give you a short overview, schematic drawing about a drug substance, a typical drug substance production overview. So it starts with a production culture, which is under sterile conditions. The cell culture, or in some cases E. coli fermentation, must be sterile to avoid any kind of failure during the cell culture or the culture propagation. The next step is the so-called harvest step, where either the E. coli cells or the CHO cells or mammalian cells are collected, and then one usually starts with the first chromatography step. If it's a monoclonal antibody, then in many cases it's a so-called protein A-based chromatography step. After that, we have the viral inactivation. That's a mandatory requirement for production of cell-based products. There must be some viral inactivation. The second chromatography step, in some cases, an ultrafiltration concentration. Again, virus filtration, a third column, a third chromatography step, and the final so-called UFDF, ultrafiltration, diafiltration, formulation step, and the conditioning of the drug substance. And steps starting from harvest up to drug substance are not sterile, but what we call it low bioburden, meaning there are multiple 0.2 micron filtration steps in between to ensure a very low bioburden load. But it's impossible to achieve sterile conditions during the so-called purification step, or everything starting from harvest up to drug substance. Things are a little bit different for a drug product. it starts, or it usually starts, with the thawing of the frozen drug substance. To remind you, drug substance production and drug product production are usually in different sites, different continents in some cases, so there must be some shipment. And this is usually done with the frozen drug substance. Some drug substances are even or just refrigerated, meaning stored at 2 to 8 degrees Celsius. But in most cases, the drug substance is frozen to ensure proper shipment and to extend the shelf life. Those tanks must be thawed, and in many cases, multiple tanks must be pooled to one drug product batch. The second step, or the step after pooling, is dilution or formulation. And there are some differences. Some companies, for example, add the final formulation here during drug product manufacturing, for example, the addition of polysorbate or certain buffer additives like amino acids. At Roche Genentech, we have a slightly different procedure. We have the final formulation step at drug substance formulation. So in our cases, we usually only filter, or in some cases dilute, or if it's a lyo product, we have a lyophilization step. After the dilution, there's the bioburden reduction filtration, again 0.2 micron filtration, and after that, the sterile filtration for fill, which is the final really sterile filtration, so that the end product, the drug product, the final drug product, is at final sterile form. And then we have the filling, stoppering, and lyophilization, capping and storage part, which is under sterile conditions. So if you look at the entire process chain, it starts from the thawing up to the bioburden reduction filtration. And again, those steps are under low bioburden conditions. It's not fully sterile, but the final steps, those are sterile. And if you look at the entire process flow, it consists of both sterile and low bioburden steps. And this is important if it comes to your microbial control strategy. So taking together, manufacturing is based on aqueous systems. In many cases, we have ambient temperatures, room temperature, or in some cases 2 to 8 degrees, and everything occurs under largely neutral pH conditions. And the process intermediates, which I have shown to you, are usually heat sensitive, meaning that filtration is the method of choice for reduction of bioburden loads, and there's no way to heat treat the process intermediates. And because of this, because of those circumstances, meaning the neutral pH, the aqueous systems, the ambient temperature, and the fact that bioburden reduction can only occur via filtration, overall, the large-scale production of biologics is prone to microbial contamination. And to ensure that there's proper control, bioburden and endotoxin occurs at defined sampling points throughout the entire GS and DP manufacturing process. And that's maybe one of the main differences between small molecule processes, meaning chemicals or peptides, and biologics. The small molecule processes usually only have a very low number of in-process testing points or sampling points, whereas biologics usually sample and test throughout the entire production process. So those so-called in-process steps are very important for biologics, in addition to the testing of the drug substance and DP, meaning that for biologics, it is not sufficient to only test drug substance and DP. You have to have additional in-process sampling points. So what's the problem now with those in-process sampling points? What needs to be considered? So first of all, endotoxins are potent pyrogens. They are ubiquitous, and so therefore monitoring throughout the manufacturing process is needed to ensure that the total endotoxin load of drug product is below the pyrogenic threshold. So if you have a monoclonal antibody and it is injected into the bloodstream, you have to have endotoxin testing in place, and you must achieve a certain or you must meet a certain limit, and this limit is based on a safety calculation. However, there's no companion or health authority guidance on setting of in-process limits for endotoxins. So there's a lot of guidance available for setting specifications for drug substance and drug product. You have guidance in USP, UP, FDA is providing guidance, but none of them provide guidance how to set in-process limits, because things are not directly linked to the safety limit. It's more related to process capability and also to method capability. When setting in-process limits for endotoxins, one has to ensure that they provide an appropriate level of control while not triggering superfluous investigation. So there's no sense in setting super tight limits, which are, for example, below your method capability, or which are simply too tight, because this would only trigger activities in your quality system which do not add quality to your product. So you have to find a balance there. And there are definitely two main challenges if we talk about in-process endotoxin limits. One is the variance in inputs from raw materials. Endotoxin will pass through filtration, the 0.2 filtration, into the process, and therefore the endotoxin load from raw materials will vary from process to process. And this is maybe one major difference between setting bioburden in-process limits and endotoxins. If there is a certain bioburden load of a raw material, this is usually filtered out. So you do not necessarily need to consider the bioburden load coming from raw materials. But if we talk about endotoxins, they easily pass the filtration, so you have to consider this. And the second point, also very different from bioburden here, and that's about the method capability. The matrix, the sample matrix, which consists of protein and has a certain pH and so on, and this sample matrix may cause inhibition or enhancement of the LL assay. And this impacts the limit of quantitation of that specific sample. And the LOQ of the endotoxin assay may vary widely between different samples and between different batches. So we all know that the LL assay, which is the standard assay for detecting endotoxins, some companies may have, for example, recombinant factor C assays in place, but this is largely the LAL-based assay. This assay, the LAL assay, is prone to inhibition and is impacted by the sample matrix. So you will see this variability. And those two aspects, the variance from input from raw materials and the method capability, do have an impact on the overall setting and the process to achieve endotoxin in-process limits. And I will come to this in more detail later. So what is the procedure for setting endotoxin in-process limits? Some general considerations and requirements I would like to give here. And especially I want to talk about provisional, what we call provisional limits. So in general, endotoxin in-process control comprises alert levels and action limits. So we have a two-tiered system. However, for products with less than 30 data points per sample, we install a so-called provisional action limit of greater than 3.0 EU per mil. And we don't define an alert level. Why? The reason for this is that 30 data points are needed to do any meaningful statistical calculations. If a certain product does not provide this number, you cannot really calculate a limit. So therefore, we decided to establish this, what we call generic provisional action limit, for every product which has less than 30 data points. And this is, for example, for IMP or IND batches, meaning batches which are produced for clinical trials. They usually run in campaign mode, and usually you have less than 30 data points. So this rule here applies to nearly all of our clinical products. And the limit per se, this value here, 3.0, was derived from historical data, meaning we have screened all our IND and IMP batches and see whether we can define a meaningful limit from this. We took into consideration the LOQ and a wide range of different products, and we came up with a number slightly lower than 3.0, but we decided to use this value here because, again, it's a valid compromise between method capability and not triggering superfluous investigation. So this 3.0 is our generic provisional action limit. And this action limit applies for all in-process steps, except if the process control step is not expected to provide endotoxin clearance downstream of a given sampling point. And in such a case, the action limit at that sample point must not exceed the drug substance or drug product release criterion. And I will give you an example in the case study, and just to explain briefly what I mean here, if there is a drug substance or DP specification of, let's say, 1 EU per mil, and you define an action, or you want to define an action limit for the very last process step, you have to take into consideration whether there's a possibility to further reduce your endotoxin load, whether there's any potential clearance. And if, for example, there's only ultrafiltration, diafiltration, most likely such a process step cannot provide endotoxin clearance. It's different if you have, for example, a chromatography step with a certain resin, and yeah, your MSAT department will tell you this can provide endotoxin clearance. So it's a case-by-case decision whether or not your final process step in drug substance manufacturing will have this 3.0 provisional limit, or will have a limit which is your drug substance or DP specification. And the process steps between drug substance and drug product, for example, the thawing and the dilution steps, they must also be considered when setting the limits between drug substance and drug product, the in-process limit. So this is about the provision limits which we apply for clinical products or products which are going to BLA. Now I would like to talk about products which are already on the market or which provide more than 30 data points. Here we can really calculate the limits. And if we have more than 30 data points, then we have our two-tiered in-process control system, meaning we have alert levels and we have action limits. Those 30 data points, they should only be obtained from GMP batches, and this is to ensure proper data quality. We do not use data from non-GMP batches like, for example, tox batches. The 30 batches used for calculation of limits should have been manufactured with different batches of raw materials. Again, to remember, we have this variability coming from the raw materials, so you should provide numbers coming, or batches coming from different raw material batches, if possible. Sometimes some raw materials, it's very difficult because they have huge batches, or the amount of raw material per batch is so low that one cannot guarantee variability or different raw material batches for each produced drug substance batch, but at least you should try to include this variability. And you should not exclude, you should not use endotoxin data coming from limit excursions, adverse trends, or secondary contaminations, because those data would erroneously increase your calculated limits. If you would include those data, your limits are simply artificially high. And regarding the alert levels, we calculate them, again, a minimum of 30 data points by determining the so-called 95th percentile. That's a known statistical approach, and if we look at the data in more detail. So you may see a lot of those data points with values below LOQ, for example, small less than 0.6 EU per mil, and in such a case, you would use the exact value, meaning 0.6 EU per mil, the LOQ, for calculation. And by the calculation of the 95 percentile should be done with an appropriate software, for example, Excel, but there are also other software solutions available. And the alert level is defined as greater than the 95 percentile. So from, let's say, a technical point of view, the calculation is easy. It's more about selecting the proper data set and considering those different raw material batches and whether or not the data come from limited excursions and have any kind of background like secondary contaminations. Now coming to the way we calculate the action limit. And this is, again, a very simple calculation. We simply multiply the endotoxin alert level by with a factor of 6. Why factor of 6? We came to this number because we considered the intrinsic method variability of the LAL assay, which is a factor of 2, that's given in the compania, and we multiplied this with a common statistical factor, which we think is, for example, 3, if you look at the 3SD variability. And we did this to achieve an action limit which is meaningfully different than the alert level, given the inherent variability of the LAL assay. Because we felt that setting an action limit very close to the alert level doesn't make any sense, and of course, the action limit should not be too high. So this number of 6 was found to be a reasonable compromise. The calculated endotoxin action limit cannot exceed the provisional action limits or action limits which have already been established. Meaning if there's an already established limit which is below the calculated one, the already established will not be changed. So you cannot increase your action limit by simply doing this calculation. Or if your calculated limit would be, for example, 3.6, you cannot implement this because your provisional limit is at 3.0. So it really depends on your specific product and your specific process whether or not you will implement this calculated limit or whether you will stay with your already established provisional limit. We defined that the minimal number of an action limit is set at greater than 0.3 EU per mil. Why? It's quite easy because we said, every endotoxin value which is below the specification of WFI, which is 0.25 rounded to 1 digit means 0.3, should be considered as safe. This is the WFI specification given in the compania, and everything below this should not be critical at all. So therefore, we decided the alert levels, yes, they can be lower, but the action limit, the minimum action limit is greater than 0.3. We do not implement action limits smaller than this. And again, considering that if a process control step is not expected to provide endotoxin clearance downstream of a given sampling point, the action limit of that sampling point should not exceed the drug substance or DP specification. And this is, again, the example with the UFDF, which, for example, cannot provide sufficient endotoxin clearance. When setting endotoxin in-process limits, it's also very important and equally important to give guidance for investigating excursions. It doesn't make any sense to implement limits without any idea of what to do if one exceeds those limits. And I don't want to go to all of those details here. It's more to provide you an example. If we have an alert level excursion, you can or cannot, that depends on your quality system, start a deviation but whether or not you do this, you should definitely review this holistically, and you should consider all the findings and gather as much as important information as possible. And we, for example, have for low-level excursion, we have 2 steps. The first step would be to conduct a laboratory investigation, meaning we have to be sure that this limit excursion is not resulting from a simple lab error. And if this has been finalized and we are sure that this is not a lab-related error, then we proceed to the next step, which is review data for an adverse trend. Meaning if there's some kind of repetition, you may escalate this to an investigation or a deviation in your track and in your quality system. So meaning that the procedure for alert level excursions is pretty straightforward. It's the most important thing is it is a warning system, meaning you should watch your process carefully for the next couple of batches. There might be the possibility that your process is running out of control, but it doesn't mean that exceeding an alert level automatically results in potential patient safety impact or critical shifting of your process. It's simply a warning. Things are different for action limits excursions because the action limit defines, if I have exceeded this, something is going wrong with my process. And so therefore, after doing the lab investigation, the same steps as for alert level excursions, you have to determine the cause. So you have to know why is this occurring. You have to find the root cause. And the second step would be evaluation of impact and assessment of the scope. So you need to know what is the impact to your to my batch. And then, of course, you have to think about disposition of batch and identification of potential kappas. And those different actions given here are very similar compared to, for example, bioburden limit excursions. Of course, adapted to the endotoxin specific requirements, but basically very similar. Now I would like to give you a case study, and I have to apologize for this table here, which is hiding the very last line of the third bullet point. This case study is taken from one of our production sites, and this biotech manufacturing process consists of CHO cell fermentation, cell harvest, and 6 protein processing or purification steps. It's, let's say, quite a unique process. It's not a standard process because usually one does not have so many protein processing steps, but in this case, for this product, we do have those different 6 different protein purification steps and 2 different cell harvest steps. The in-process alert levels were calculated from historical data sets with a minimum of 35 data points by determining the 95 percentile as described before. And if you look at the table here, the column called number of data points, you can see that the different samples of the different process steps have different numbers of data points. In some cases, more than 100, and in 2 cases, 36, in 1 case, 35. Why? It's a simple answer. We have implemented additional sampling points after commercialization, meaning during lifecycle management. So we started with a lower number of in-process sampling points and added additional sampling points based on our microbial control risk assessment. We decided to add those sampling points, and that's why we have this or those different numbers of data points. The action limits were calculated from unrounded 95 percentiles by multiplying with a factor of 6. As described before, that's our standard calculation for action limits. And the calculated alert levels were rounded to meaningful values, meaning 1 digit after, for example, 0.1. And the calculated action limits were rounded to greater than 0.3 EU per mil if the values were less than 0.3 EU per mole. And again, this is because of the WFI specification and our approach to not install limits below 0.3. And if you go to the different samples or processing steps and look at the table, you can see a column called calculated limits. You can see the values for the alert levels. You can see the values, the calculated limits, action limits. And on the right side, you can see the column called applied limits. And if you look at the alert levels, you can see we used the rounded values. So meaning for cell harvest step number 1, we have a calculated value of greater than 0.022. We rounded this to greater than 0.1. For the second harvest step, we have a calculated value of greater than 0.096, rounded to 0.1. Processing step number 1, greater than 0.320, rounded to 0.3, and so on. And if you look at the action limits, you can see that the cell harvest step 1 and 2 resulted in very low calculated action limits, but the applied limits or the implemented limits were higher, so greater than 1.9. And the reason for this is, if you look at protein processing step number 1, you can see that the action limit, the calculated action limit, is greater than 1.9. And we decided to use this higher value also for the cell harvest steps, meaning those process steps further upstream of the first protein processing step. And this is because limits applied to earlier steps should not be tighter than steps further downstream. So we usually follow this approach an approach where samples or process steps further upstream usually have higher limits compared to those processing steps further downstream. And this rule was applied here, meaning that we have 3 processing steps with an action limit of greater than 1.9, and then we used the rounded values in case of processing step number 2. The calculated value is 0.48, rounded to 0.5. And for all the remaining steps, 3, 4, 5, and 6, we applied this rule of not having an action limit below 0.3. The meaning is that you start with an action limit of greater than 1.9, you go further down to 0.5, and then finally to 0.3, meaning there's a decrease of your action limit if you go further downstream. And this example is a case study where the product is produced at only one site. Things are a bit more complicated if one has to consider multiple sites. For example, if your product is produced in different continents or at different production sites, you have to ensure that you have uniform action limits. That's simply a health authority expectation that you should have uniform, harmonized action limits for a given product, and you can usually not have site-specific action limits. So the calculation basically is the same, but you have to consider additional aspects, like, for example, LOQ at the different testing sites. And those aspects are really case by case. So it is recommended. It's not a strict requirement, but it is recommended that you apply the same method at the different testing sites if you want to install a uniform harmonized action limit for a given product. And you should also try to really come to the same LOQ whenever possible. It's difficult because it would mean that your testing is really harmonized. You need to agree on, let's say, the main or main factors of your endotoxin testing, and this is something which is not that easy to achieve, but those aspects are important because if you have production sites which have extremely different LOQs, it gets very difficult to calculate an endotoxin in-process limit because you have simply very different data sets. If you are able to achieve this kind of harmonization, or if you have a single site product, things are really easy. And I think the approach which we have developed here at Roche provides us this statistically justifiable approach. It's based on historical data, meaning that we consider the process capability, simply what can the process provide in regarding endotoxin clearance. And it also ensures appropriate responses are taken in if the endotoxin limit is exceeded. And also very important, we find procedures in case of limit excursions, meaning a clear guidance what to do, what needs to be considered if a limit is exceeded. We have a two-tiered system for products with more than 30 data points, meaning we have an early warning of drifts in the process. That's why we have the alert level, and we can also detect serious microbial control failure, which triggers actions and clearly defines the path forward regarding batch assessment or whether or not the batch can be used or can be released. I would like to acknowledge two of my colleagues. Jens Westendorf, he was providing the case study data, he did all the calculations, and my colleague Michael Knight and the entire Roche Genentech Endotoxin Technical Forum team for the development of the concept. It took us nearly 2 years to develop this and to agree on the procedure. We have a lot of different sites being involved in this process, and we had to meet all the different requirements. And now we have a robust process in place. We received some FDA feedback last year, very positive feedback. So it looks like this approach can be seen as, let's say, accepted by health authorities, or at least we did not receive any negative feedback here. By saying this, I finalize, and I would like to hand over to Ursula.
Thank you so much, Friedrich. Now we'll begin the question and answer segment of the webinar. Once again, I would like to remind the audience that you can still send your questions via the questions tab on the right-hand side of your screen. In the meantime, I just would like to inform our viewers that Biopharma Asia will have planned webinars on topics similar to this. So please visit our website at www.biopharma-asia.com and go to the resources tab at the top for a list of all upcoming webinars. The upcoming similar webinar will be presented by Karen Zink-McCullough of MMI Associates and Kevin L. Williams of Biomerieux. The webinar will call Preview of USP's Informational Chapter Guidelines on the Endotoxins Test, and it will be held on the 11th of June. So if the audience hasn't got any questions, I would like to ask Friedrich if you have any closing remarks you can share with us now.
Yeah, my closing remark might be that setting up in-process limits for biologics is a complex procedure, not regarding the calculation, but more about the data collection. And one has to be careful to take to pick the right data sets. And I can only emphasize the need of harmonization between different production sites, especially for the multi-site products. It will definitely help and facilitate the procedure. And I think harmonization beyond what is given in the pharmacopoeia is definitely something which is recommended, not only for this process, but also for other aspects. And I can see, yeah, let's say some kind of trend regarding this, that sites and different production sites tend to harmonize more and more. And there's additional guidance, for example, for standard curves. There's additional guidance for sample variability, CD values. This is not given in the Compania. That is recommended by the appropriate SMEs, and companies should really consider this as an important aspect, this harmonization and standardization process.
Thank you so much, Friedrich. Again, for more information and for more webinars, please visit biopharma-asia.com. Thank you so much, everyone.